So thank you all again and welcome to the session. I'm honored and privileged to be co-hosting this session of the International IVF Initiative, again about SARS-CoV-2 in assisted reproduction. Just some housekeeping first. So for the videos of the previous session, please go to ivfmeeting.com and if you can, just leave a review, a positive one, of course. And I encourage you to ask all your questions using the Q&A session of the conference service and not the chat function. So the housekeeping is done. My name is Filippo Zambelli, and it's indeed a great honor to be co-moderating my session with Dr. Ilan Tur-Kaspa and with our special guest tonight, Professor Alan Parker.
Thank you, Filippo. I'm very pleased to be hosting this session with you. It's a pleasure to introduce Filippo Zambelli, who graduated in biotechnology from the University of Bologna and received a joint PhD from the University of Teramo and the Free University of Brussels. His early studies focused on human embryonic stem cell differentiation, and then he developed a special interest in the genetic makeup of embryonic stem cells, oocytes, and embryo. After his PhD, he joins a basic research lab in Clinica Eugene, where he is currently working as a staff scientist, mainly focusing on interplay between mitochondria and transcriptional changes in oocytes. It's also a great pleasure to have with us once more Professor Alan Parker, who actually was part of the first meeting. He is a virus tinkerer on social media, and he has a long-standing interest in biology and how to apply to cancer therapy using oncolytic adenovirus-based vectors. Lucky for us, his team in Cardiff University has switched to work on COVID-19 to try to assist to solve this pandemic misery and to try to get us some understanding better of this new virus that has gripped the world. So, and you can look at both CVs on our websites, and you can impressed as I am from both of them.
So now it's my great pleasure to introduce my co-moderator that will also give the first talk tonight or today, depending on where you are, guys. So Ilan Tur-Kaspa, is a medical doctor, is of course an internationally known specialist in reproductive medicine and infertility. He's the founder and medical director of the Institute for Human Reproduction in Chicago and Oak Brook, Illinois. And is the chair of reproductive imaging, a special interest group of the ASRM. He's author of lots of peer-reviewed scientific papers, over 110, and also book chapters, and is also co-editor on the books of ultrasound imaging in reproductive medicine and biotechnology of human reproduction. Dr. Tur-Kaspa also served as a professor at the Ray Division, Department of Obstetric and Gynecology at the University of Chicago, and is the president of the Chicago Association of Reproductive Endocrinologists. So he will give us some insight from his paper, COVID-19 may affect male infertility, but it's not sexually transmitted, a systematic rate. So the talk will be recorded, despite the professor is here. So enjoy.
Thank you for the kind introduction. It's hello, everybody. My name is Ilan Tur-Kaspa, and I'm from the Institute for Human Reproduction in Chicago. It's a pleasure to share with you today our recent publication, a systematic review about COVID-19 may affect male fertility, but is not sexually transmitted. I would like to thank my co-authors, Dr. David Cohen is my partner, and Grace and Tomer, who were joining us as interns during the summer and winter of 2020. And yes, Tomer is my son, and this was really a pleasure to have him. They're both, Tomer and Grace are pre-med. It was a pleasure to have him as part of this project. There are no conflict of interest related to this presentation, and whoever wants to ask any questions or later on ask for reprint, here's my email. I'll be happy to share it. So SARS-CoV-2 has led to a fast-spreading global pandemic of COVID-19. By December 11, 2020, this is the date that we have closed our review, it infected over 71 million people worldwide and 1.5 million deaths globally. From a public health perspective and infertility treatment, it is crucial to determine if SARS-CoV-2 is sexually transmitted. To cause a sexually transmitted disease, a virus has to be detected in seminal or vaginal fluids from asymptomatic or symptomatic people and be transmitted through intercourse or insemination or IVF. Since the putative receptor for the virus, ACE2, is identified in the productive organs, it was important also to examine if COVID-19 may affect human fertility. So the objectives of our systematic review was, one, to determine if SARS-CoV-2 is sexually transmitted, and two, to examine if COVID-19 may affect human fertility. Now you can see here a picture of the virus. The red is a strand of RNA surrounding by protein capsule. The S spikes, the non-S spikes, that gives them the name of the corona. And this is like a gene machine. Actually, it's a very tiny, it's 100 nanometers, which you can see only with electron microscopy. And once it attached to the ACE2 receptors, and you need also a specific protease, TRAMPSR2, to get it involved too, together in about 10 minutes, the virus can penetrate into the host cell within about 10 minutes into the cell, which is 100 times larger than it. So we performed a systematic review of global epidemiological investigation, molecular receptor identification analysis, and detection studies of SARS-CoV-2 in the male and female reproductive tract by December 11, 2020. We revealed, the systematic search revealed about close to 2,000 publications, of which 70 studies were included in this review based on relevance, including 23 studies which tested the male and female reproductive tract of 404 adult COVID-19 patients. So altogether, in 2020, by December 11, there's only 404 patients that actually were tested for possible transmission in the male or female reproductive tract. This is a busy slide, but however, I specifically wanted you to see that because it actually represents the global information on SARS-CoV-2 in seminal fluid of COVID-19 patients. And initially, in March, April 2020, there were a few publications demonstrating no virus particles in the seminal fluid. However, in May of 2020, there was a publication in JAMA by Li et al. From Shanghai that actually suggested that in 16% of the 38 men that they tested, so it's about 6 men, they actually found positive viral particle, RNA particles in the seminal fluid. They concluded, and we all agree with them, that if their finding will be confirmed by others, then this may be, then the virus actually can be transmitted sexually. This is a huge turn point with regards to what should we do from public health perspective, as well as for what should we do at our lab. And if you remember, this was just a time in early May that actually in the US and many other places in the world, IVF unit came back to start treating patients after a shutdown for about 6 weeks from mid-March to end of April. So altogether, when we looked at all the studies of 299 men, actually you see that this was the only outlier. Only one paper found 6 men with positive particles, RNA particles in the seminal fluid. Everybody else, it was negative. So 98%, it was negative. So altogether, based on this data, and based on, we agree with the authors themselves that say that if nobody else find it, there may be an issue with that. There was some, with specific to these papers, there was also some methodology concerns that the authors needed to provide some more information later on. When we look at the testis, as well as prostatic fluid, you can see here the prostatic fluid. In 89 men, prostatic fluid was tested in 3 different studies. All were negative. When we look at testicular biopsies, this is all from men who died from COVID-19, who passed away, and they did testicular biopsies. And in one, there was a positive test for SARS-CoV-2. Nevertheless, the authors themselves say that they think that this was a contamination from blood, and actually the virus itself could not be identified by electron microscopy. So let's move to the female reproductive tract. And here we have some studies. 4 studies looked at 58 women in vaginal fluids, in cervical smears, in pap smears, as well as in oocytes. And if you look here, while in male, we had most of the data came from asymptomatics or mild or moderate patients. In female, most of the data came from severe, from women that had severe COVID-19. And still, there was only 1 case report that found COVID-19 in vaginal fluid. All cervical smears were negative, and in publications that came from Spain, in 2 donors that actually found to be positive for COVID-19, but they were asymptomatic. 16 oocytes from those 2 donors were actually tested. They tested the oocytes themselves. They were all negative. This case that was positive in vaginal fluid was actually a 67-year-old woman who was diagnosed by 2 tests to be negative, then 2 tests to be positive, and then another test to be negative, all within days. And there was no correlation to the clinical situation. So one can say, you know, the patient can be negative initially, and then the disease to be more severe can be positive, and then can be negative again. But there was no correlation to the clinical, as well as some methodological issues. So again, we as well as others think that there is no data to support that SARS-CoV-2 may be sexually transmitted disease. Now, we looked for evidence for impact of COVID-19 on fertility. Now, it is not known if or how COVID-19 might affect female fertility, ovarian endocrine function, or ovarian reserve. There was a publication in March in Molecular Human Reproduction suggesting that because the receptor is found in the ovary, it's going to be a major effect on female fertility. However, there's no data whatsoever that published that support this hypothesis. Now, COVID-19 might affect male fertility by damaging endocrine function and/or by temporary or permanent changes caused by elevated body temperature and or orchitis. Elevated body temperature and orchitis are well-known cause for diminished male fertility. Now, evidence of damaged endocrine function was demonstrated by a significantly lower serum testosterone to LH ratios, which is a measure used as a predictor of compromised testicular function, and it was found in COVID-19 patients compared to control. Now, there were 2 studies that actually investigated semen analysis in patients recovering from COVID-19, 8 to 54 days after diagnosis. One demonstrated a decreased sperm counts and motility compared to controls. The other study found that sperm count, motility, and morphology to be within normal range. So here we are, this is a global data by December 2020. Now, what happened in the testis of patients with COVID-19? We have the virus. Does it cross the blood testicular barrier? And then what will be the role of the ACE receptor and the TMPRSS2 receptors, as I told you, the proteins, and what it will cause, and what might be the mechanism that is going on. Some suggested even maybe vitamin D involved in it too. Now, it's really important to see whether the TMPRSS modulatory protein that is needed for the required for the virus to enter the cells, except that it's attached to the receptor itself, the ACE2. So this is a crucial point, I think, in the understanding why actually it may not, the situation is not as bad as we thought it might be. The ACE receptor, ACE2 receptor is found in reproductive organs. There's no questions about it. However, most of the recent studies actually suggest there's a lack of co-expression of TRPS2 modularity protein, which, as I said, this is the one that's required, that after the virus attached to the receptor, it needs this modularity protein in order to penetrate the cell. And we don't find it practically in testicular cells, sperm, or oocytes. And actually, this rejects the hypothesis that gametes may transmit SARS-CoV-2. Now, local inflammatory damage of the testis-blood barrier, it's possible. There was a survey of about 90 patients that were hospitalized for COVID-19. 11% of them had testicular pain. However, when they did a clinical evaluation, there was only one that can be diagnosed by epididymal orchitis. A very nice study performed with using ultrasound imaging criteria for orchitis or epididymal orchitis. And actually they found that in patients younger than 40 years old, only about 6% had ultrasound imaging criteria for orchitis, compared to 25% in patients older than 80 years old. So, and they also correlated to the severity of the disease. So the more severe COVID-19 can be, or the older the man is, the higher the chance that he may suffer from orchitis. Now, another two studies actually show 7 men who demonstrated orchitis-like symptoms. However, they all tested negative for SARS-CoV-2 in the seminal fluid. To conclude, no epidemiological investigation of SARS-CoV-2 in tested seminal prostatics and vaginal fluids and cervical smears have confirmed that COVID-19 is an STD. So yes, there were some few studies that actually throw a stone in the water. However, we think, as well as others now, that it is not an STD. Now, COVID-19 may have temporary or permanent determinantal effect on male reproduction, as it may involve endocrine alteration in LH and testosterone, decreased sperm parameter, and may induce an inflammatory orchitis, resulting in fibrosis and further loss of testicular function. Now, this information is important for clinicians, for guidelines for health recommendation, FDA, ASRM, ESHRE guidelines for gamete and tissue donor eligibility, and of course for fertility treatment and regular laboratory precautions. Universal precautions, practiced in clinical setting and in the art laboratories worldwide to prevent transmission of known or unknown viral infections seem adequate and sufficient at that time. Further prospective and longitudinal control studies are needed to investigate the potential effect of COVID-19 on human fertility. Thank you very much.
Okay, so thank you very much, Ilan, for your very nice talk and very clear message, let's say. So I'll go first with a question, and I will leave the difficult ones for later, but just more a general question that I really like from James Duffy from the UK. So thanks you for the excellent presentation, of course. But he asks, with the rapid publication of case series, how easy it is to distinct individual cases, report series, and ensure they do not include participants which overlap with others? So are we sure we're reporting a single patient all the time, or are we reporting same patient most of the times?
So this is a great question, but this is a good question for any scientific data that is published. I think that during the first 3 months of the pandemic, a lot of the data that was published was whoever sent publication, everybody was eager to have some information. So it was easy to get in. Just to let you know that, for instance, this paper by Lee at JAMA that was published in May. So we actually did our first, we done our meta-analysis. We actually done it in July, and we sent it to JAMA and said, you published this paper, you have to publish now a systematic review saying this is not an STD. They said, no, we have too many. We already have too many publications on COVID-19. All right, so just to show you how things move, but I think it's excellent. It's also really important, and when you do a systematic review and meta-analysis, you really need to read those papers carefully and make sure that you count whoever comes in, what patients are in, what patients aren't, and really, and if we actually in our systematic review, we also graded the level of evidence, and actually all those publications are between 3 to 5. So the level of evidence, whatever is available, but this is actually the global data that we had at that time. Now I think some more and more publication we may see soon. We may see some recent data that came again, which supports the same conclusion.
Yeah, sure. It was probably a problem that we have in general, but the short timing made it even worse. So yeah, we needed to know something, and indeed, maybe at the beginning was not super controlled, let's say.
That's right.
Anyway,
That's right.
Thank you very much. But let's move to the next talk. So we will have an exciting Q&A later on. And so again, it's my pleasure to introduce Manuel Viotti. So you know him, and you saw him already not long ago talking about mosaicism in the very same conference. So Manuel Viotti is a senior scientist at the Zambelli Foundation for Reproductive Medicine. He has a special focus on molecular and cell biology of embryos and uterine environments. He had a PhD in developmental biology from Weill Cornell Graduate School of Medical Sciences and the Sloan Kettering Institute from New York. And then he joined the biotechnology company Genentech from San Francisco, California, as a postdoc. He then became involved with the establishment of the Zambelli Foundation, so again from California, San Francisco, Foster City, in 2017, and since then his work has featured several papers in fertility, human reproduction, RBMO again, and many others. But again, I'm not saying anything you don't know already. So he also engages in clinical embryology and PGTA, and he holds several accreditations. So Manuel, the floor is yours.
Thank you so much, Filippo, for that kind introduction. And thank you for the organizers, of course, for inviting me to talk. My name is Manuel Viotti from the Zambelli Foundation, and I have nothing to disclose. So obviously, we all know COVID-19, and we all know SARS-CoV-2 by now. And we are familiar with the most common symptoms, which are the fever, the cough, shortness of breath, and fatigue, which can obviously, as we know, lead to death. But sometimes we see other symptoms that are not related to the lung. For example, liver dysfunction, gastrointestinal problems, skin changes, eye problems, and neurological complications, and others as well. And it's not immediately clear whether this is due to a direct infection of those organs by SARS-CoV-2, or it's a secondary effect due to a systemic infection and a systemic problem like inflammation or immune system related. But it's important to know that the viral tropism, which is kind of the range of tissues and cells in the human body that SARS-CoV-2 can infect, that is currently being defined right now. What we do know is obviously that it can spread very effectively between people by horizontal transmission through droplets that we exhale, but it's very unlikely that it can be transmitted sexually, as Dr. Tur-Kaspa just presented, or at least there's very, very little, if any, evidence for that. But what I'm going to be talking about here is another type of transmission, which is the vertical transmission. Basically, that it's between generations from the parents to a developing embryo or fetus. And you might have seen this headline, which made a big splash. And this was a baby that was infected with coronavirus in the womb. And it's a really interesting case. Where in March 2020 in Paris, in France, a patient was admitted at 35 weeks of gestation with fever and severe cough. And they did the test, and the patient was positive for SARS-CoV-2. And after 3 days in the hospital, they observed category 3 fetal heart tracing. And so they did a C-section. And the baby tested positive for SARS-CoV-2 as well with nasopharyngeal swab. This is actually the original, the original report on that, what that headline was based on. And you can see, what they observed was inflammation in the brain of the neonate, which is consistent with what's sometimes observed in the adult as well. And they also saw inflammation in the placenta and molecular evidence of infection of SARS-CoV-2 in the placenta. So what the authors surmised was that the mother had passed the virus to the fetus through the placenta. And luckily, though, both mother and baby are recovered and are doing well. And after this, other reports followed in the literature. And in an effort to estimate how often this happens, Cotlier and colleagues performed this very thorough meta-analysis where they summarized all the tests that had been done in newborns from mothers with COVID-19 in the 3rd trimester. And the most common test that was done was nasopharyngeal swabs, and in 936 cases, they found 27 had a positive SARS-CoV-2 test. So from this, they surmised that during the 3rd trimester, if the patient gets infected with SARS-CoV-2, there's a 3.2% chance that the patient passes it to the fetus. But importantly, there was no significant morbidity or mortality to the newborns, which is good news, of course. In that same review and meta-analysis, Cotlier and I'll discuss these recurrent patterns that observed with other viruses of vertical transmission. Basically, with increasing gestational age, so as the pregnancy progresses, there's a higher incidence of vertical transmission. Although there's a lower incidence of detrimental effects on the fetus. And conversely, earlier in the gestation, there's very low incidence of transmission, but if it does happen, then the effects, the medical effects on the fetus or the embryo can be severe. And then they go on to state that we could assume that COVID-19 may also have a similar detrimental effect when maternal infection occurs early in the gestation. And that begs the question, obviously, can human embryos even become infected by SARS-CoV-2? And now I'm going to present some data from a study that we're conducting, trying to answer this question. I just need to quickly review the mechanism of entry of SARS-CoV-2 infection, which depends on the interaction of the spike protein on the surface of SARS-CoV-2 with 2 factors located on the host cell membrane. First, the spike protein engages the receptor ACE2, and then the spike protein gets cleaved by the protease TMPRSS2. So when these 2 steps are complete, then the virus is internalized and infection occurs. So to know if embryos can become infected by SARS-CoV-2, the very first clue is to check whether cells of the embryo express the 2 factors ACE2 and TMPRSS2. So we started our study by doing a transcript level analysis by RNA-seq. We focused on the trophectoderm since it comprises the outer layer of the embryo and therefore the primary access site for to potential viral infection. And we looked at 24 blastocysts, and for each one we collected a 5 to 10 cell T biopsy and processed it for RNA-seq. And these violin plots display expression levels, and each dot represents 1 embryo. And they confirmed that the transcripts of both ACE2 and TMPRSS2 were present in the samples. To then investigate if ACE2 is present on a protein level as well and where it is localized, we performed immunofluorescence and confocal microscopy on blastocysts. And the left column shows a projection image of a blastocyst with nuclei in blue and ACE2 in white. The middle column shows ACE2 signal alone in white. And the third column shows a high magnification image of trophectoderm cells. And we observed enrichment of ACE2 between neighboring trophectoderm cells as shown by the pink arrowheads, which is consistent with localization on the cell membrane. And that's exactly where one would expect ACE2, if it's a functional ACE2 receptor. And by the way, this confirms a previously published study that reported the same observation using a different commercially available antibody raised for ACE2. So we confirmed that observation. For TMPRSS2, there was signal in T cells with a more general distribution that we saw with ACE2, but it was still present on cell membranes as indicated by the pink arrowhead. So taken together, this data suggests that the necessary factors are present in cells of the embryo, specifically in the trophectoderm, which is surely a prerequisite for cell entry. But can the virus actually enter the cells? And one way to explore this question is using reporter virions. And this is an image of a regular SARS-CoV-2 virion. And this is a virion based on a different virus that has had its cell entry proteins removed. And so it cannot infect cells by itself. And it also encodes a green fluorescent protein in its genome. So one can engineer a reporter virus by taking the spike protein of SARS-CoV-2 and sticking it on the membrane of the reporter virion. And this process is known as pseudotyping. So the pseudotyped reporter virus will only enter the cell if there is functional interaction between spike protein and ACE2. So if this happens, the virion is internalized, the genetic material is released and translated, and the infected cell turns green. And so by monitoring which cells show green fluorescence, we can identify those that are permissive to the spike protein and therefore to SARS-CoV-2. And this is the experimental design for which we obtained IRB approval. We started with embryos that had been obviously donated to research. And at the blastocyst stage, those that had not already hatched, we mechanically removed from their zonas. And that's an important point since the zona might add an extra layer of protection. And we wanted to see just if trophectoderm cells can be infected at all. Although obviously embryos do hatch at some point, so that layer of protection is removed even in the natural situation. The embryos were then spinoculated with a reporter virus, and after some extra incubation time, they were imaged by confocal microscopy or by bright field microscopy with epifluorescence with GFP. And here are some sample confocal images of our results showing nuclei in blue. So each one is obviously one embryo. The nuclei are in blue and GFPs in green. And these embryos clearly show numerous GFP positive cells with robust cytoplasmic signal indicating successful viral entry. So these are just representative images. And this is kind of now a summary of the results. So first, we used negative controls. So we just took the embryos through the process without adding the pseudotype virus. And as expected, 15 embryos, we did not observe any displaying GFP fluorescence, green fluorescence. The same was the case with the reporter virus that had not been pseudotyped. In other words, the bald virus. So no embryos display fluorescence as expected. However, when the spike protein was present, we saw fluorescent cells in 6 out of 15 embryos. And one can then use neutralizing antibodies targeting spike, which interfere with it and block its interaction with ACE2. So when we added the neutralizing antibody to the mix, we saw a reduction of infected embryos, and there was only one case with punctate or weaker GFP signal. So this reporter virus was based on HIV, on the HIV virus. We then repeated the experiments with a different virus based on the VSV virus. And here again, with the negative controls, we saw no fluorescence. But when we spiked it with the SARS-CoV-2 spike protein, we observed GFP positive cells in some of the embryos. When we added to the mix blocking antibodies towards ACE2 or TMPRSS2, we saw a reduction of infected embryos. The 2 embryos that show fluorescence had just very limited puncta. And then as a control for that, we added just an anti-IgG neutralizing antibody, and there we saw the fluorescence again. We then repeated the experiment with pseudotyping the virions with the spike protein from SARS-CoV-1. And there we saw fluorescence again. And SARS-CoV-1 also enters cells via ACE2. But then when we did it with MERS, with the spike protein from MERS, that we did not see fluorescence. And MERS, in fact, enters cells through a different receptor, which is DPP4. So this directly implicated ACE2 and TMPRSS2 in this process of cell entry. So from this data indicates that preimplantation embryos are permissive to SARS-CoV-2 in the trophectoderm through the canonical spike ACE2 entry mechanism. And obviously, even though we've addressed that specific question, this finding really raises many more questions. For example, to what extent can the viral life cycle progress within those cells? So we know, or we have good evidence now that the virus can enter trophectoderm cells, but can it complete the viral life cycle and release new virions to the environment, for example, and infect neighboring cells? By the way, we also mined our RNA-seq data for factors involved in downstream steps of SARS-CoV-2 life cycle, and such as replication and viral assembly and trafficking. And we saw that many are present in trophectoderm cells. So that might suggest that the viral life cycle can progress further than just entry. Another question, obviously, is what would be the effect on the cells and the embryo as a whole, and by extension on the pregnancy. So if we plot the spectrum of all potential consequences, it ranges from having absolutely no effect to have severe detrimental effects and lead possibly to cell death and embryo death, which would result in failed implantation. But the effect could also be somewhere in the middle, and which could arguably even worse. So if the embryo does not perish immediately, but experiences long-term effects that cause things like miscarriages, placental issues, or congenital abnormalities. I mean, this is obviously very speculative, of course, but I think the mere notion that the virus can enter trophectoderm cells should put us on guard, and if we're not already on guard, obviously. And another question is obviously, are there realistic scenarios that bring together the virus with preimplantation embryos? And we've already heard about some of these studies from Dr. Tur-Kaspa, this study from Virotherapies group, where they didn't see SARS-CoV-2 in oocytes of infected females. There's plenty of data showing that the vast majority of samples from sperm and semen of infected males do not have SARS-CoV-2 present. Although, as was already discussed, there's that one study that showed that there can be, and obviously, so then maybe we cannot dismiss this possibility altogether yet. The other potential is that the virus gets brought to that region through microlesions, through the blood and microlesions during intercourse in natural conceptions. And then in the art clinic, there are additional avenues of exposure. For example, through infected personnel and through breathing and coughing, can the virus fall on dishes or some other way access the embryos that are grown in the clinics? And well, again, this is very speculative, of course, but I think it's still worth considering when discussing the best practices in the clinic during the COVID-19 pandemic. And this is something the next speaker, Dr. Swain, will discuss. So with all these questions, obviously more research is needed, and I'm really happy to report that just a few days ago, we found out that we were awarded a research grant by ESHRE that will let us continue this project and answer some of the burning questions. And with this, I just want to thank obviously everybody that was involved. This project brought together embryologists and virologists, two groups of people that don't often interact, and so that was very interesting and rewarding. And obviously, thank you very much for listening.
Manuel, thank you so much. It's a fascinating lecture. Always a pleasure to hear about all your great research. I have a few questions, and I'll get you a few one of Fatima and Mitch, both of them. So they ask, first of all, will the zona pellucida can or not be a barrier for infection, and whether what happens if the virus infect blastocysts? First of all, where would it come from? And second, would the blastocyst can now infect the mother? So how would you think about that?
Okay, So to the first question, can the zona be an extra barrier? I'm not sure we can definitely say one way or another. You know, when you look at the size of the virus and the size of the pores in the zona, it's kind of tricky to really make a conclusive statement. But anyway, you know, when at some point the blastocyst has to hatch even, you know, during pregnancy, or like to achieve pregnancy. So that extra protection will be lost at some point, and that's where maybe the highest risk of exposure takes place. So I don't know the answer to that question, but those are kind of, to the other questions, you know, it's I briefly went over it. You know, are there real scenarios where the embryos can be exposed to the virus? It's on speculation, but maybe through blood of infected patients, like I said, through microlesions during sexual intercourse, and maybe that one study supporting the seminal fluid notion, but it's only one study, like you discussed. I don't know, and then in the art clinic, like I said, maybe there are other avenues that we haven't even thought of. So it's very speculative. Again, this finding actually asked more questions than it answers. We took a reductionist approach and just answered that one question: that can in theory it even enter the cells of the embryo? And I think we've answered that it can. Although what happens next is just we don't know, and it's for us to find out soon.
Thank you so much. We will come back for you with a session with a Q&A. I'm sure that there will be a lot of questions. It is a pleasure. Thank you. It is a pleasure now for me to introduce you to Dr. Jason Swain. Jason Swain is a PhD, HCLD. And he is currently have a very prestigious job. He is a corporate laboratory director of the CCRM IVF Laboratory Network, which is overseeing a growing network of 11 clinical IVF laboratories in North America. And you all know about CCRM, so Jason, congratulations. It's a great lab. He completed his PhD in molecular and integrative physiology at the University of Michigan. And he is responsible now for CCRM laboratory design, protocol, procedure implementation, as well as staff training and ongoing quality control monitoring for the network and biology, andrology, and endocrinology laboratories, and the CCRM donor egg bank. His bio is even his short bio is too long for me to read it all, and I'm sure that you will be enjoying reading it on the website. And it is a really pleasure to introduce to you Dr. Jason Swain for our next speaker. Please, Jason.
Thank you very much. I'm going to be talking about the SARS-CoV-2 virus, really within the four walls of the IVF lab, and maybe touch upon some of those questions that Manuel just had and nicely kind of deferred a little to me in terms of, you know, some possible real world scenarios or not in terms of exposure to our gametes and embryos in the lab. And most of us are aware of the variety of these sort of commentary papers from early last year when we were all struggling with how to deal with this pandemic and whether there was risk in our labs and perhaps more importantly, if there were things that we could do to address these risks and still safely practice and try to get our patients pregnant. And a lot of these were overly cautious for obvious reasons. But as we've gathered more data, you know, the hope is that that data will then help guide us to continue practice safely. And so really, you know, the big concern or topic of this talk is virus really inside the IVF lab. And I'm going to talk more about, you know, potential sample to staff infection or between samples in terms of cross-contamination. And these are going to be the two main areas. And you know, we all know that we have existing viruses that we deal with on a daily basis in our labs, HIV, CMV, Zika, a variety of others that we don't screen for. And so really, I guess the question is, are there specific or unique concerns with COVID-19 other than it being an airborne virus? Does it bring something else unique with it that causes us to treat things differently than we do already with things like HIV and hepatitis. And I won't spend too much time on this, but I think we've now very clearly demonstrated that it does not appear that the virus is present in semen. There is the one paper that identified it, but several that did not identify it. And we just heard Ilan very nicely summarize all this data in much more detail, where it doesn't appear to be an STD, at least from these papers. And we'll all, again, continue to monitor research in the literature to see if that changes. But these sort of papers prompted us to do an examination in our lab, and we started this probably back in May or June of last year, and wanted to gather some data to see, is the virus in the lab? Is there a real world exposure risk, as was previously asked by one of the attendees? And so we gathered some data quickly and submitted it and presented it at ASRM of last year, and then we subsequently published this recently in RBM Online. And so this is really going to be a very quick summary of this particular data. And the majority of this work was done by Sandeep and Dr. Khan here in our research facilities in Denver, Colorado. And so, just briefly, we used our safe treatment approach that we employed when we reopened for business and started treating patients. And that really simply was testing all females coming through with nasal swab PCR testing approximately 3 to 5 days before retrieval, looking for a negative test. If it wasn't negative, the procedure would be canceled. We did not use PCR testing for men because they weren't undergoing anesthesia, and there is a cost aspect to this, but we did do screening with questionnaires, and all men were asymptomatic who were donating their samples. And then importantly, all semen was prepared using a basic double layer gradient 40-80 followed by a couple washes and then a swim up with ICSI. And so that's our normal protocol. We did not take any additional precautions in terms of specialized inserts or things like that. And then we analyzed 3 main samples in our laboratory. We looked at follicular fluid and around 300 samples there. We looked at vitrification solutions, and this was because it's the last dilution step before the embryos would be placed into liquid nitrogen storage tanks. And so we wanted to look at that last dilution to see if there was a potential exposure risk once placed into the tanks. And then we also looked at culture media. This is where the embryos spend the majority of their time. If there is potentially going to be exposure with hatching and things that Manuel talked about, it'd have the most likelihood to be exposed during this extended time in culture media. So we looked at our blastocyst culture media on 61 samples. They were all processed slightly differently based on volume and other aspects. And then we did a multiplex qPCR assay looking at 3 different loci. And we did have positive and negative controls. And so, tried to make this as sensitive and complete as possible to avoid any false positives or false negatives. And really kind of cutting to the chase, looking at, these 300, 261 samples of these various media, we were not able to identify virus in any of these. And so that's reassuring that at least with our screening and our protocols of not putting through any known active patients, that the virus was not readily apparent within the 4 walls of the IVF lab. And so I do want to be clear that this does not mean that virus cannot be present in the media. We did not treat any known infected women, and men were untested, but asymptomatic. And we did not test individual sperm to see if perhaps, those may be carrying the virus. And obviously, this was done on a limited number of samples. So I would say that based on these findings, as well as some of the others that were already talked about, and then I'll review here very briefly, it does seem unlikely that this would end up in the lab, at least if you have some semblance of safety screening in place. And that comes from, these other 2 papers that were referenced by the earlier lecturers. This is undetectable viral RNA and oocytes from positive women. This is just a couple women as noted previously. And then here's another one, failure to detect viral RNA in follicular fluid from a SARS-CoV positive woman. So certainly not high numbers, but does add to a growing body of literature saying that it does not appear that virus is present in the lab thus far. And I don't know that this is too surprising. This is a well-known paper that's been out there now for several years from Anna Cobo and her colleagues, where they looked at known STDs, things that we know we need to be concerned about in the lab, hepatitis B and C and HIV. And they looked at, 3 follicular fluid samples, 20 samples of media, various numbers of embryo culture and LN2 samples, and they weren't able to detect these viruses either. So I would say that our findings, kind of go hand in hand with some of these previous findings in terms of the relative risks or presence or lack thereof of virus in the IVF lab. This is a kind of a must read on this topic, I would say, from Kim Pomeroy and colleagues, talking about the storage of preserved tissues in reproductive labs and evidence of cross-contamination being negligible. And they go through, a variety of rationale for this. And really kind of the take-home point is there's a lot of dilution that goes on. And, it is difficult to identify virus in the lab with the dilution, with the safety protocols that most of us already have in place. Perhaps more timely is this paper from Dr. Shiwi and Dr. Pomeroy talking about cryopreservation in the time of COVID-19. And so they touch on some of those prior points from that prior publication, but do mention some things that might be more relevant that have already been touched on here in terms of the zona pellucida barrier and how that's breached more and more these days with biopsy and blastocyst culture, but also, potential preventative measures such as closed vitrification devices. And, there are some implications on this in terms of lab outcome and certainly lab training and validation. If we're adopting new technologies to try to prevent an infection, we want to make sure that our outcomes are as good as the previous device. And in this case, many vitrification devices are open. But if the viral concern isn't that large of a concern, do we then undertake some of these more labor intensive or yet to be validated approaches? So food for thought. And it is something that we do need to think about and continue studying, as we just heard from Manuel. The receptors and machinery are present in the blastocyst, and others are showing these on gametes as well. So we do want to be vigilant. We do want to do more research, and we want to see, how real of a risk is this, because certainly we do not want to be the cause of some of those negative outcomes that we'd heard about from the other lecturers. And so then kind of, wrapping things up in terms of COVID-19 and any specialized or added concerns in terms of liquid nitrogen storage in our labs, it is apparent that pathogens can bind gametes and embryos. The receptors are there. There is a zona pellucida, but that gets removed at some point or is breached at some point, as noted. But however, the pathogens don't appear to be present in the fluid, at least to date, with the studies to date that we're seeing in the laboratory. Mixed results in regards to semen, follicular fluid. As we said, blood is kind of the outlier there. Is that a factor or not? Blood was present in some of our samples in our study. We still didn't identify the virus, albeit in women who had tested negative right before transfer. And then, as I mentioned, dilution is a common theme here when you read some of those prior reviews and get involved in some of these discussions in terms of the risk in the lab. That being said, we do have some recommendations from the various groups that help oversee our field. Here's one from the first update of ASRM, where it's recommended that samples from known infected patients be handled in the same manner as tissues from patients who are seropositive for other infectious diseases. I think most people are comfortable with this. If you've got a policy of not treating patients who are infected, it becomes, I guess, less of an issue. But if they are known to be infected, you would isolate them just as you would a hepatitis or HIV patient. This is a more recent publication that come out in February from ESHRA, where they've got the recommendation of, because viruses can survive and can potentially be transmitted via liquid nitrogen, that you would separate the storage of the reproductive cells according to the viral positive or negative status. That would mean that you'd have to be testing your patients. And again, I don't think anyone would argue with this. This one perhaps is a bit more interesting or intriguing. Separate cryopreservation dewars to be used to quarantine with unknown infectious status. Again, if the virus hasn't been identified yet, what is the real risk? We don't know. If you're not testing, you're now mixing patients of potentially known positive and known negative. And again, going back to how is this different than the other viruses that we deal with? If you're routinely already putting patients and combining potential positive and negative in tanks, maybe you're okay setting up new tanks, but this really only protects your new patients or patients moving forward and not necessarily your historical patients. And so, just a couple of, I guess, comments or rhetorical questions that maybe prompt some Q&A at the end of this lecture is, does the use of separate tanks at this time make sense? Does it matter if we're talking about eggs, embryos, or semen? Is there a greater or less risk with one of these than the other? And as we said, if you don't have testing in place to confirm positive or negative, that poses an interesting question. And this is a potential thing for the field. Do we test media? Do we implement testing in the lab to see if the virus is there in terms of trying to determine how to store some of these? Is there a risk of cross-contamination? And again, a point here is that if somebody's known positive, many recommend delaying treatment. So does this become less of an issue or not? And certainly there's cost and space considerations when we do talk about some of these specialized storage approaches. And so with that, I will turn things back over to our moderators.
Thank you, Jason, for an excellent presentation. Go ahead, Filippo.
Thank you. So before passing on to the Q&A, I'll ask you a quick question. And the question is from Kelly Tilleman from Ghent University. So she's one of the main, let's say, one of the coordinator of the special interest group of quality and safety in ART. So it's a person that's very well informed. And she asks you, why only screening at the time of pickup? Why not before transfer, especially when we talk about day 5 transfer? Shall we do it? Why should we not do it?
Yeah, so for the purpose of this study, it was within the 4 walls of the IVF lab. We don't really do many fresh transfers. Most of our transfers are frozen. So we do additional screening prior to transfer to make sure that we're not bringing COVID positive patients into the clinic. But for the purpose of this study, we wanted to look at the follicular fluid, culture media, and the vitrification solutions. And so obviously testing the patients prior to retrieval for the purposes of this study is what made sense. But practically speaking, not related to this particular study, we do test women before they come back for frozen embryo transfer as well.
Okay, so yeah, but not for the day 5, you just test once because it's very low the possibility, of course.
Exactly, and we're more concerned about the risk, for anesthesia and again, the samples coming into the lab.
Absolutely. And so I'll say we can move on to the Q&A session after we got all these different perspectives. So is the virus in gametes? Can it enter embryos and can it come from the lab? So we can start the question and answer. If you want to start, Ilan, you have some questions?
Sure. But I am actually very encouraged to see that all of us actually, while we have to be cautious, of course, there can always be something new. But as it's my clinical understanding, that actually, for now, there is no actually data to support that this is an STD. So this is important because actually, the diagnosis of STD has to come from epidemiological studies. Somebody has to have a disease somewhere, travel somewhere, and while having intercourse or having IVF should transmit the disease. There is no data to support that. So now we have to be precautions. And of course, this is important. But the universal precautions that we are currently doing, since Jason and everybody else that we talk, we agree that actually, if you keep those, this is sufficient for now. Even the FDA actually updated the guidelines for donor screening and came out in January 4th, 2021, just 3 months ago, and said that actually they don't think that they should revise the guidelines with regards to SARS-CoV-2. The only thing they do recommend that, of course, if somebody is sick now, then if you can wait 28 days, 14 to 28 days, this would be lovely. So from donor perspective, if the donor had a disease, you can wait actually 28 days and she can go for egg donation. And there's nothing to change in the protocol of what you do with regards to sperm donor or egg donation, at least from the FDA point of view, as of January 2021. So I just wanted to, if Jason or Manuel, do you have any, do you agree with this conclusion or not?
No, you mentioned egg donation, just an interesting, I mean, it's maybe anecdotal, but we also do surveillance testing in our lab. Every patient that comes through and staff as well, we do indoor SARS-CoV-2 testing. And since we started it last May, the only cases that have been positive, there's been 5 cases positive, they were all egg donors. And I don't know if this is like it's just anecdotal, but it's just it was, since you mentioned it, that popped into my head. Yeah, I don't know if Dr. Swain has something to add, but.
And I think it's encouraging. I mean, we do in-house testing. We adopted that early on, try to be as safe as possible. We've had, various positive tests. We've canceled procedures. I think that's the hard one for people. But we, especially with multiple labs, did set a policy early on and get agreement that if somebody did test positive, that we would stop treatment to avoid, the variety of issues, whether it be in the lab or just staff safety or even patient safety with anesthesia, since we use anesthesia during retrieval. And an interesting thing that comes up is some of these, I guess, chronic patients that test positive, but then continue to test positive. So that's kind of a conundrum too, where we want to bring patients back, and so we're now starting to see some of these where they've recovered, but they're still technically testing positive with our testing. And so we are trying to evolve as the situation evolves and make sure that we can still safely treat these patients and not, keep declining them care, because it's a hard enough decision to make in the first place when somebody gets all ready to go and you say, oh, we're not going to go to retrieval, you're sick, get better. Most people are okay with that, but then months later to still do that based on, these positive tests that are lingering, it's something that we're trying to stay ahead of, because again, we don't want to end up with an issue in the lab or something even worse down the road.
Right. Ellen, would you like to answer this last comment by Jason?
I mean, the thing I'd say, I take a very different perspective on it, from being a virologist rather than embryologist, and I would simply look at the evidence out there in nature that viruses have a route of transmission, and you don't see any other viruses out there that are both respiratory and sexually transmitted. And they tend to have a tropism, they tend to have a route of transmission that they use, and this is very clearly a respiratory virus. So the fact that we're not seeing transmission sexually or reproductive infection is probably not a surprise. It's what we should be expecting here. We've obviously been trying, or you guys have been trying very hard to see it, and I think that's evidenced by the literature. Ilan, you've shown the evidence from all the papers that have shown that there is potentially viral transmission in semen. There's one paper that has shown that it has been, of course, got a lot of press, which is often the way when you find something unexpected. But I think you're probably right. I think the likelihood is that is likely to have been cross-contamination from that study. So I think it's something we have to be wary of, we have to look out for, but I think it would be extremely unlikely that we're going to find a virus such as a coronavirus that's going to be both respiratory and sexually transmitted.
Thank you, Ellen. There's some another question, maybe Manuel. There was a question by again Mitch and Fatima. They looked about if there is a vertical transmission or not.
Yeah, we were discussing this earlier. Well, in the 3rd trimester, again, there's that meta-analysis that suggests that it might happen with very low frequency. They came up with this number 3.2% based on what's been published. It's a low percent. So again, are we talking about contamination? Are we talking about bad results? On the flip side of the argument, somebody could say, are there a lot of false negatives that we're not finding? And if the test is bad, maybe there's more positives that we're not finding. So clearly I understand what Dr. Parker is saying. Usually a virus is actually sexually transmitted or transmitted through I don't know the technical term, like through breathing or
respiratory blood or
respiratory. But vertical transmission is still possible. You know, the one does not preclude the other, so it can pass horizontally through respiratory, but then can vertical transmission still happen through the body? It's tricky. Like it's. I don't know if we can very clearly say no. I think there's some evidence that it might happen. And like we saw, there's other viruses that where it does happen. The Zika cytomegalovirus. When does it happen with this one? I think the jury is still out, according to my understanding.
So for all those models, or for all those different viruses, there are animal models that you can test that hypothesis. I know they have limitations, but I don't know. You may know better than I do whether any animal model has shown transmission vertically in pregnant animal models, or if you could use your pseudotyped viruses, in fact, to test that. I mean, it's the one thing with that approach I caution slightly is, of course, a pseudotyped virus is a pseudotyped virus. It's not the actual virus, of course. And of course, you need an animal model that's at least semi-permissive of viral replication, which mice tend not to be. But I guess if you used a pregnant ferret or something like that, would that provide a suitable model that you could infect via the respiratory pathway and then see whether that virus is reaching the placenta, the developing placenta, and the fetus?
Yeah, I think that would kind of add, contribute evidence to it. To me, again, that study by the Lucas group is probably the most convincing one that it does happen occasionally. But yeah, we're still at the tip of the iceberg here, I think. And it's hard to get to those questions. I mean, we're talking about human embryos or human, you can use animal models and then draw conclusions from there, but then does that really translate to the human condition? There's always that caveat as well. I think we don't have the answer yet.
And we don't necessarily have all the models to test them either, which is the frustrating thing too as well, right? So many limitations.
This is why we need great scientists. Filippo, you want to take it from here?
Oh, yeah, sure. Actually, I was just wanting to add that probably also our testing and our way of obtaining samples in these cases is not yet at the probably scientific excellence, because, again, we were in a situation that the first reports were not really possible to control properly because we didn't know what it was. So yeah, this probably adds to our uncertainty. Anyways, Manuel, I wanted to ask you something on following up this topic, and it's a question from Fernando Prados from Brazil, Sao Paulo. And how many particles, how many virions were added to the embryos for your assays? Was it mimicking a natural, possibly natural infection, or was it also like a lab accident that you?
No, that's a great question, because if we try to think of what is the natural level of the number of viruses that an embryo would be exposed to, again, it's completely speculative. We don't know if that even exists in nature, whether that exposure even exists. And if it does exist, what would it be? And so it was hard for us to mimic the real situation because we just simply don't know if it even exists at all. I guess the very basic question we were just asking is like, are cells of the embryo, some cells of the embryo, do they even have the machinery available to internalize the virus? But now the translation from that to the real situation and to physiological situation, it's a big jump that we're not gonna follow up and try to do that, but we're still at, like I said, tip of the iceberg there.
Absolutely. And also there is a whole different side of this problem. And I'm going to go for another question very, very relevant from Kelly Tilleman for Alan. And we as art people are so concerned, while not one viral expert stated that people could not procreate at home. So are we over-worrying? Are we making too much of a fuss?
Sorry, I'm just reading the question again. As arts people, assisted reproduction, right? Okay, you can tell I'm not an embryologist, right? Okay. I'm not used to your acronyms.
Maybe we're over-worrying ourselves, while the natural thing that happens all the time, and many orders of magnitude more, it's not a problem.
So this is it. You know, I'm not in the reproductive field, so I don't spend a lot of time thinking about these kind of models. You know, the bigger question obviously is, you know, what was the potential for transmission by the respiratory route, which is the predominant route that's being used here. But of course, there's other, you know, is blood, does that carry virus? It doesn't seem to, you know. There are many different ways that viruses can transmit, and ruling them all in or out to, you know, niche areas for each different field. I don't think anyone should underestimate, you know, the importance of these things. Obviously, it's important that they're investigated, and the job you guys do, you know, you have to not only think about, you know, natural reproduction and sexual transmission, but also, like you say, in the lab. You know, that's an eye-opening thing for me to learn about, and the measures you had to put in place in the labs to mitigate any accidents, cross-contamination, all those kind of things is mind-blowing, because they're the sort of things that I would never think of. But you know, that in my field, we are thinking about gene therapy vectors and these kind of things. We think about how viruses can be manipulated and so on. And you know, this is why I'm thinking about for the vertical transmission question that Manuel's looking at. You know, what model could you best use for that? In the field I work in, viruses can be used as gene therapy applications where you treat in utero. You know, that's a big field to try and correct genetic mutations. So there are ways of testing, if you put the virus into close contact in the pregnant animal, whether there's then integration then, and that will answer some of these questions as a sort of halfway house between the in vitro assay, where you're culturing your embryos in the dish and putting a huge amount of virus on, which is possibly completely artificial unless there's some horrendous accident in the lab. It's a sort of halfway house, that more relevant model, I guess. But the short answer is no, I don't think you're underestimating it. I think it's important that we do consider all these different aspects, and that every different area of medicine and science is doing so.
Yeah, indeed. So let's go for maybe another question for Jason. For example, Estrella Rosenberg from Caracas asks, do you label the cryopreserved embryo with special labels? Like, shall we just keep track of them?
Yeah, so I mean, that's if we did treat positive patients, we would treat it as reactive. You know, here in the U.S., you know, per ASR recommendations and prior FDA guidelines, we would put them in a positive known tank or in isolation conditions and label them as such. But if we are not testing, there's no way for us to label it, and we're not, you know, trying to set some date in the sand, you know, of March 1st or December 20th or whatever, in terms of when did we think we may have had COVID exposed patients and start, you know, new tanks over here. We haven't done that. I know others can. And again, it's hard to fault people for trying to take a safe approach. So, you know, the short answer is no, we currently are not labeling anything different because we're not treating known positive patients.
Yeah, also probably when did it come to our labs before we started testing is a very good question, because it was not when we started, it was probably from before. Yeah.
No, I think that we started testing patients from May of 2020, and actually until like September, October, everyone was negative. So while we reevaluated and maybe thought to stop testing, we started to have some positive with the new wave of the winter that started. And I see Jason saying yes. I probably saw the same. And Manuel, no, we didn't have donors positive. Actually, they were all patients. So I don't know whether it's coincidence or not, but definitely if we take your clinic and our clinic together, now we have half and half. So here, just to show you that we had all donors were negative actually, and patients were positive. And yes, we canceled the cycle because of all the complexity of everything, and just tell them wait 28 days and come back. And this is by just to follow guidelines with the CDC, etc., and FDA, just to be on the precautious side. None of us wants to be a cavalier and says, "Oh, there's no vertical transmission," or the people may find it. There may be some data to suggest certain things, etc. But from clinical point of view, from the world point of view, is this a major issue? Yes. If it was a major issue, we would have seen all those cases already, and we don't see it yet. So is it possible that we'll have some maybe with the new variants of the disease, you can see certain different behavior? It's possible. And I think that Manuel, your studies is amazing and fascinating. What its relevance to the clinical world now? For now, nothing. We just know that yes, if you take embryos out of the zona pellucida, can you get to mimic like the possible potential infected? Yes, it is.
Yeah, although it'll be hard. Obviously, of the cases where they, at least the few cases where there was evidence of vertical transmission during the 3rd trimester, all the babies, or pretty much all the babies, were recovered and are fine, and the mothers recovered and are fine. On the other hand, if early vertical transmission happens, and we see things like failed implantation, obviously in natural conceptions, failed implantation because of vertical transmission is going to be very, very hard to perceive in in numbers of analyses. But the main concern would be something like there is vertical transmission that happens occasionally, say like an infected mother, infected father, conceived naturally, and somehow the virus gets into that region where the embryo develops, infects the embryo implants, and then something happens to the cells of the trophectoderm that causes restricted placental growth, or so on and so forth. Obviously, it's very speculative, but I'm just saying there is where that might manifest itself over time, with especially in areas of the world with high incidence of SARS-CoV-2. We might that will kind of the data will come through if we see the higher numbers of miscarriages or so on. Maybe it's too early to tell yet. But in general, I guess we're a field that wants to be conservative and as safe as possible. So when it comes to things like, should we vaccinate pregnant women? So depending on who you ask, even some regulatory bodies, they still disagree on that. Which pregnant women should be vaccinated? And how about people that are planning to conceive? Should they vaccinate or not? I think these are questions that play into those questions. I mean, these are observations that play into those questions.
What would you recommend them, if I may ask?
I think so. I mean, okay, so with the highest risk, as far as I can tell, of getting the vaccine as a pregnant woman is the fever that develops at the 2nd dose of Moderna and Pfizer in 35% of cases or so. The high fever that can then affect the pregnancy. So that's something to keep in mind. That's the biggest risk. On the other hand, if we're now talking about potential vertical transmission, even though it's not, like you said, it's not a universal occurrence, but even the potential of it, increases, in my mind, the risk-benefit analysis of whether to take the vaccine or not.
Maybe
Ellen,
we can ask
yes.
take on it.
Ellen, you knew we are going to get to use this question, so don't drop it on us back to us. Don't say, this is a hot potatoes. Now it's on your hands.
So my understanding is that there are obviously more reservations around vaccinating pregnant women, but the advice is that they can still be vaccinated, although it's with a certain degree more caution. And I haven't heard of any major adverse effects in terms of loss of miscarriages being induced or anything like that. So my hunch would be that it would be the right thing to do. And if you're talking vertical transmission, the situation that's going to be, I guess, most dangerous in the case of the fetus isn't just going to get SARS-CoV-2, so the mother's not going to have it. They're both going to have it at the same time. So the antibodies that are raised will be useful for both mother and fetus developing. So yeah, it's right that these things are very carefully trialed, but my opinion would be that the vaccine should be offered to pregnant women. I was going to actually ask Manuel, in terms of the studies you showed were really elegant with the pseudotype virus and the very compelling data showing that you're getting some GFP expression in those. You hinted at those studies maybe suggesting that the virus can get in and express its transgene, but may or may not be able to form a replicative infection. So are there studies ongoing in your lab or in others with the full SARS-CoV-2 virus to see what happens to those embryos, whether there is viral replication in there, whether there's any, issues with the embryos, what's happening to them? Are those studies happening?
Yeah, I don't know of other labs doing it. I know we're now working with the live virus.
Yeah.
Initially because always we're confined with embryos donated to research. Like, so if you initially we said, should we throw the live virus on the embryos, and what will we observe? Maybe nothing, but then can we actually like exclude anything? Because we only have 48 hours of development of the embryo in the dish, and then it perishes. Obviously, we could have seen an effect, but we decided to go this other route of the pseudotype to a reductionist approach again. Just saying, can it even enter the cell? And now we're going to that experiment, to those experiments you're mentioning. If we now throw the live virus on the embryos, do we see any effect? Do we see cell death? Do we see anything that would make us think that might have a real consequence, a clinical consequence? Yeah.
And of course, what you also showed there was that your neutralizing serum or your antibodies, whatever it was, purified antibodies or neutralizing serum, were protecting those embryos. So maybe that's an advert for vaccinating pregnant women, right?
I think so. And my understanding is even later in pregnancy, the protective antibodies that the women develop pass to the fetus. So that's another argument for vaccinating during pregnancy.
Right. And I think, Manuel, you correctly say that we may not have seen some of the things that you're looking for, maybe miscarriages, etc. But you have to remember that already 100 million people were sick with the disease. And hopefully we are going to get most of the people in the world vaccinated, and maybe we are out of the pandemic. So if we didn't see it by now, maybe we won't see it later. But there are some questions from the audience with regards to COVID-19 and men infertility. They are related to from Brownstone and Fran Fairley. With regards to do they affect male fertility, whether they affect, or maybe it's because of the temperature that we know may has effect on fertility. Jason, these questions were to me and to you. Would you like to start?
Yeah. So in terms of the viral effect on male fertility, was that the question?
Yes. What do you think? What is your opinion?
Yeah. So I think my takeaway was that it's likely the symptoms or the fever, the sickness that's causing the decrease in semen or sperm counts, things like that, not necessarily the virus acting directly on the sperm cells. Your presentation focused a lot on this. So, the question then comes up, and I'm sure it's probably listed, is if you then wait 3 months for the new spermatogenic wave, will you see, the sperm count come back up? And so it'll be interesting when those studies come out. But then, the next question is, what about sperm quality? Is there DNA fragmentation? And if there are some defects on sperm quality other than just count, if you wait your 3 months or so, will we see a reversal of some of those possible detriments? So I guess that's, stay tuned to be determined. But from what I've seen, it seems more like the fever or the temperature, as you indicated, are likely the main issues at this point.
Yeah, I agree with you. And another question I wanted to ask you is, would you think, if it's possible that the sperm may transmit SARS-CoV-2, do you think that ICSI may prevent it?
So I don't know that ICSI would prevent it. I mean, I think Mitch Shuey and others
reduce the risk. Yes, reduce. Yeah,
so may say that that's now going to introduce it directly into the egg and potentially cause an issue if there is an issue to be had. So again, we didn't measure whether virus was present, on the surface of sperm. Through our processing, our dilution, it would have presumably been removed from any seminal fluid. And then again, you get down to numbers of, viral particles and things like that. So another great question in an area of research, I think, yet to be conducted, because ICSI could pose a potential risk. In our case, we did it as part of our normal routine because we do a lot of genetic analysis. So we didn't modify our protocols really at all for this. This was just kind of our standard, and we do ICSI on the vast majority of our cases, and some would argue too many cases. But if people were to show virus on the outside of sperm, would standard insemination potentially be better? I don't know, because now you may not have as much of a dilution effect, where you're not picking up an individual sperm that's been further diluted in PVP, one at a time in an ICSI needle. So, I think the fact that the majority of these studies have not shown it in semen is what I tend to focus on. And so if it's not in the semen, or if it's not in the semen sample, what's the likelihood of it being attached to the outside of sperm? So I think that's what it comes down to. And at this point, as you pointed out, it doesn't seem to be widespread, at least from those 7 or 8 studies, minus the one that showed 6 patients.
Right. Filippo, you want to continue?
I think it's about time to wrap up. So I think we all agree that the situation is not so dramatic as it might have been when this whole thing started, but we still need some time to figure out the things properly, just as a general wrap up. But I have a very important question for Alan, actually, and feel free not to answer if you don't want to. But when you watch rugby, who do you cheer for? England, Wales, or Scotland?
That's a very dangerous and naughty question.
Feel free not to answer.
Honestly, I cheer for England because I'm an Englishman living in Wales, but I will lose a lot of friends now around the local region, I suspect. But I cheer very quietly. But I cheer for all that. And actually, to be honest, I lived for 7 years in Scotland before moving to Wales 8 years ago. So I'm a bit English, a bit Scottish, and a bit Welsh. And the kids were born in Scotland and have been raised in Wales. So we cheer for all the home nations. Honest.
Okay. Nice. Very polite and balanced answer. Nice. Okay, so jokes aside, it's time to wrap up. So I want to thank you, everybody. So everybody, all the participants for taking time to participate, and of course all the speakers. So my co-host and speaker Ilan Tur-Kaspa, Manuel Viotti, Jason Swain, and of course our guest star Professor Alan Parker. And thank you for the organizing committee that made this possible. So Jacques Cohen, Peter Nagy, Giles Palmer, Thomas Elliott, Sheishta Sadruin, Marianne Sheveititz, sorry. And also today's Q&A team. Thanks a lot again for Sheishta Sadruin, Marianne Sheveititz, and Anna McLaughlin.
Thank you all. It's been a pleasure. We thank all the I3 group and Peter Nagy and Giles Palmer for all his support. This session and presentation will be available on the website soon. Don't forget to rate our lecturers. It's very easy. And your certificate of attendance will be sent before commencement for the next session. Our next session is on Tuesday, as always, at 3 p.m. Central Time, 8 p.m. UK, 9 p.m. Central European, the same time as always. The next session, of course, it's continuing. The world has changed. So now is it 000 or out of the office? Remote management of fertility clinic, what it's going to be. Is it going to be back to the old days? Is it going to be hybrid, or just continue as it is now? And it's going to have videos from all around the world, for most remote clinics in the world, and talks on labs management by Matthew Vermilli and Michael Tykel and Charlie Kenny, with moderators of Simon Fischel and Michelle Pergini, sorry. We have one last video for you all before we leave. Thank you all and stay safe.
Thank you. Bye.
Hello, everyone. My name is Yolanda and I'm from Portugal. I'm just finished my master's degree in biotechnology of human assisted reproduction. If you want to comment on this session, please leave a review on the website www.heyvfmeeting.com. Please follow us on our social media like Instagram, Facebook, Twitter, and LinkedIn. If you have any topic which you would like to see on this webinar, then please send an email. Bye. Thank you very much.